everyone. Elizabeth Schneider, Senior Global Marketing Manager here at NanoString. We're here at the last poster that we're walking through for the day at the morning poster session, the last day of the AACR. So this will be our last Nano News break for AACR 2022, but we will see you at next year's AACR in 2023. So I'm here today with Xiang Li. He's a Senior Scientist, Scientific Affairs at Neogenomics. So he's going to be talking to us today about comprehensive characterization of biology of inflammatory bowel disease associated colorectal cancer using the GeoMx Cancer Transcriptome Atlas panel. Thank you so much, Sean. So could you start by giving us a little background about Neogenomics? What do you guys do?
Absolutely. So Neogenomics is US's largest oncology CRO, and we partner with a lot of different sponsors like NanoString and Illumina, etc. And we offer a wide variety of services, including IHC, FISH, flow, and also molecular. And we're really excited to partner with NanoString on DSP as well as their Encounter instruments to provide the best available service for clients in pharmaceutical or in industry.
Excellent. So can you briefly walk us through the study? So what kind of samples were you looking at, and what type of GeoMx assay did you use?
Yeah, so we're looking at colorectal disease. So we have inflammatory bowel disease, ulcerative colitis, Crohn's disease, etc. And we took about 20 FFPE samples that included CRC, UC, CD, and also healthy patients. And we basically ran it through the GeoMx DSP workflow, taking these FFPE, staining them with pan-CK, CD45, as well as cyto13 IHC, it's traditional IHC and fluorescent antibody markers to identify which ROIs we wanted to select. And then we also applied the CTA, or Cancer Transcriptome Atlas panel, composed of 1,800 genes, to really survey, within either the epithelial cell region or proximal stroma regions, so regions that are CD45 positive, that are really close to the epithelial cell region, as well as 4 ROIs that are in the distal stromas. And so doing this comprehensive gene profiling analysis with spatial context using DSP, we were able to identify, do a lot of clustering within the ROIs that we selected. We did heat map to really identify any gene profile patterns. And then from our volcano plot, actually, very surprisingly, we found that there were few markers that were CRC positive and highly expressed that were differentiated from either healthy patients or other Crohn's disease. And so one of the markers that we're really excited to follow up is SOX9, that it's present in basically all of the CRC. Yeah, it's very highly expressed, and we're really excited to do that. And we're trying to also complement multi-omics at Neogenomics to follow up and get sort of the single cell co-expression resolution. So a lot of follow-up studies here, and this is a very preliminary study. We had very few limited time to analyze the data, but we're going to do a more comprehensive analysis, and maybe we'll have an update on that for a future conference. But we're really excited to use this really powerful technique to survey the landscape for these cancer genes and get an overall information for this study.
So what is the next step for this? I know that you use the Cancer Transcriptome Atlas, but as you know, GeoMx now has the ability to profile the whole transcriptome. So is that something you might be doing in a different study?
Absolutely. We're very interested in doing a whole transcriptome atlas. So this, using the CTA is also part of our initial validation of the workflow, the RNA workflow, and we have a lot of interest from across the industry to use the whole transcriptome atlas, and we'll definitely be using that for different studies, and maybe even follow up with the same colorectal cancer samples for WTA. And And then so the follow-up for this, identify where the markers and then do additional studies to confirm those.
Excellent. Yeah, and another thing I noticed too is like for these proximal stroma regions, you know, you could have actually segmented between, you know, CD45 positive and pan-CK. So that's, you know, something else that you could do. So are you guys excited about the cosmic spatial molecular imagers?
Yeah,
You can
we
do,
are.
like single cell RNA and protein profiling?
Yeah, we are, and you can get really high resolution there. You can really high plex to 1,000 genes, and yeah, that would be an awesome instrument to do a study in parallel, taking the same slide and apply both. And the nice thing about DSP is that you can reuse the tissue, and so it's non-destructive, so you can apply that in 2 different instruments and get the same answer.
Yeah. So in general, it sounds like at Neogenomics, you've had a lot of customers who are really interested in spatial biology.
Yeah, we do, yeah. It's a really exciting field that we're moving forward into, and at this conference, there's so many spatial transcriptomics instruments and technology, and we have a lot of interest. So definitely moving towards the space, and really excited to working with you guys in the future.
Excellent. Well, thank you so much, Sean. It was my pleasure to be here and interview you.
Thank you so much, Elizabeth.
And I hope if anyone out there is looking for a CRO, definitely reach out to Neogenomics. They're a great partner, and we've certainly enjoyed having you as a NanoString customer.
Thank you so much.
Yeah.